Okay, that's good. All right. Good afternoon, good morning, good evening, whatever is for you in your time zone at the moment. I'm very excited for this last joint seminars in neuroscience at UCLA. And as you know, this is co-sponsored by the Brain Research Institute, the Semel Institute, and the neuroscience theme at UCLA. Today, we have 4 new members of the Brain Research Institute. They're not all new members at UCLA. In fact, all of them have been at UCLA for a little bit. And I feel a little sheepish giving the introduction here. I see Tom Carmichael on it, and as chair of the neurology department here really should be giving the introduction, it seems. 3 out of the 4 today's speakers are from neurology. So you probably know them much better than I do, Tom. So we're going to start with Ahmet Arac, who is in the, as I just said, is in the Department of Neurology. He's an assistant professor at the moment. He got his MD in Ankara in Turkey, and he can pronounce the name of the institution of the university himself. He, during that time, did summer research, both first at UCLA and then at Harvard. And then he came to the United States after that to do a postdoc at Stanford, working with Gary Steinberg. For many of you, it will be interesting to note that he actually co-published with Ben Barres, who's one of the heroes for many of us, I think. He then came back to UCLA, I guess one could say, to do his residencies and internships, did a fellowship in Rehabilitation, and during that time, scientifically was working with Tom Carmichael. He then arose to the rank of assistant professor, and he's going to talk to us today about Grab That Bottle, explaining motor control. And you'll understand what the title means after his talk. Ahmet, please. So short, it's hard to sort of determine, should we have more speakers, or should we make the talks longer? So we got that for a 15-minute time slot this time. So the next speaker is Ranmal Samarasinghe, and he got his BA from Swarthmore College, then moved to Pittsburgh for an MD PhD, did his PhD together with Don DeFranco. Then came to UCLA, where he did a lot of his internships and fellowships, and during that time was working with Ben Novich as a postdoc. And his talk is titled Modeling Neural Circuit Formation and Function with Human Brain Organoids. Ranmal.
Thanks. Can I get screen sharing permission, please?
I hope so. You should have it.
Got it.
Maria?
Got it.
Okay. Thank you.
Okay, thank you for that introduction and for this opportunity. It's really great to be able to talk to this audience and also kind of see some of the other exciting stuff that's going on. My interest, the interest of my new lab is really to understand neural circuit formation and function using stem cell models. And by formation, I mean development, and by function, I'm really talking about neurophysiology. So, of course, this is complicated to model this, and without belaboring the point, for 2 reasons, at least in the way that I think about it. One has to do with the fact that brain, and human brain in particular, is complex, and it's large, and it's anatomically complicated. And so if you want to understand the development of circuits in the human brain in particular, we need to be able to develop models that can recapitulate at least some aspects of this complex anatomy, including multiple layers, including a number of unique cell types, as an example. But the brain, especially if we're interested in function, is not just anatomically inert entity, it's also physiologically active. So this is a human example of an EEG from a patient, and I'm a clinical neurophysiologist. And so we think about the brain not just in terms of anatomy, but in terms of electrical function. And of course, an EEG is still a simplification of the sophisticated activity that happens in the brain. But at the same time, it's important because it provides some important information about the state, for example, of brain health and disease. And if we're trying to model this, we're trying to model development and function, we'd ideally have a system where we can see both functional complexity and anatomical complexity. So how have we gone about trying to address this and answer these questions? So the way I've been trained with Ben and with others is really to think as a reductionist. And so sort of the overall approach is a reductionist one, where you take this big problem and we break it down into a simple problem, and try and understand in some granular detail what's going on in the smaller problem, with the hope of extrapolating to what we're trying to understand, which is circuit function and development. So with that sort of really brief background, I can talk, the rest of the talk will really be about the basis of my laboratory and how I've approached this. And a lot of this work, of course, is my postdoctoral work, which was, as just mentioned, done in Ben's lab. So part one of this is developing model, and I'm basically devoted and I'm all in my lab with using human brain organoids. And then what we've done more recently is to try and adapt some approaches to investigate this physiological activity, using existing approaches, but also adapting them. And finally, as kind of proof of principle on this, very briefly at the end, I'll kind of describe where the lab is going, but I want to tell the story as sort of constructing the edifice on which my lab is built. So first, human brain organoids. So what are these? So these are stem cell derived, as many of you know, so they can be induced pluripotent stem cells or human ESLs. And they are these three-dimensional in vitro structures. And importantly, and this is a cartoon from my colleague who was in Ben's lab, postdoc Momoko Watanabe, these organoids have within them some of the anatomical and cytoarchitectural complexity that we'd like to see in an in vitro system. And within these rosettes, which is what this arrow, this box is kind of pointing to, we see a layered organization and cell types that recapitulate human brain to some extent. And this is just more of the same, but if we take a cross section of a certain kind of organoid, you can see a layered architecture from progenitor, PAX6, all the way to more mature neurons above. But of course, I'm interested not just in anatomy, but in function. And so we had to work to develop systems to also elicit complex activity. And of course, we needed to know if that even happens. So how do we approach this? And how am I approaching it in my lab? So fundamentally, this is the approach that we took in Ben's lab, inspired by work from Sergio Pascal at Stanford, where we essentially make certain kinds of organoids initially. So we fate specify early. So in this case, for example, we have cortex or CX, which is excitatory predominant organoid, and then GE or ganglionic eminence, which is more inhibitory predominant organoid. And we combine these to make these what we call fusion structures. They're also called assembloids in the literature. This is an illustration of us doing this. The ganglionic eminence for the GE was pre-labeled with TD tomato, and you can see the red in both sides. And so the cells migrated as we'd expect in normal development. And fixated on this a little bit because what we thought was important if we're looking for oscillation is that we have a nice balance of excitation and inhibition. And so we need to have inhibitory cells in the excitatory, in the cortex component. And when we did some immunostaining, we did see that GAD is a marker for interneurons, that we're seeing a percentage, 25 to 30%, which is what we'd see in vivo as well in human brain. And this is more recent single cell data, but we were pleased to see that our fusion organoids have upper layer neurons, deep layer neurons, interneurons, as well as other cell types that we'd expect to see and hope to see in a relatively complex in vitro structure. So what are the techniques that I'm using? So there's basically two. One is multiphoton-based calcium indicator imaging, and we had to adapt these with the help of colleagues at UCLA to extract data. So this is a live cell imaging, live organoid imaging of an organoid that's loaded with, that's infected with AAV GCamp. And so the flashing cell is a calcium indicator. And then we had to come up with some algorithms to extract this data in a consistent manner. And then with collaborators in Istvan Modi's lab, we also wanted to look at oscillatory activity. So we make the organoids and then we put in microelectrodes. And you'll see this kind of figure repeatedly in my talk, but essentially it's what you're seeing on the x-axis is frequency in hertz and the power of the frequency. And we're seeing at multiple frequencies within these organoids peaks of activity. And it's not that different from equivalent data taken from humans. So now we have the tool, which are these LFPs and calcium indicator imaging, and the organoid that we want to use, which is this fusion structure. And the next question is, can we use these techniques to see or to understand more about human disease? And so what we did was study a disease called Rett syndrome. We picked this because it's early onset, which is good for organoids. It's associated with epilepsy, which is my clinical interest. And there isn't really overt anatomical derangement in many of these patients. There's some reports of microcephaly, but it's not significant. And so I'll just start the videos. And so we had two patients. I'm only going to show you one. We have an isogenic control, so matched control, and a mutant. And if you watch these videos, I hope it shows up at least decently on Zoom. In the mutant case, what we're seeing are these flashes of activity. And I'll show you one sort of right there. And it's illustrated here in the red boxes, dotted boxes. These are the individual activity of neurons, whereas in the isogenic control, it's more decorrelated activity. And here's some quantification of the data. But essentially, our conclusion from this was that these patient organoids show this hypersynchronous activity, consistent potentially with like an epileptiform-like abnormality. So we did the same thing now using the local field potential recordings. And I think the easiest way to illustrate is to show the mutant side by side with the isogenic control. So same patient, isogenic control mutant. In the control case, you're seeing these nice oscillations. This is a blown up part of the local field potential recording on top, and you see nice oscillations. In this periodogram, in this figure, you're seeing the oscillations between 0 and 80 hertz nicely. You're seeing peaks down here. If you look at the mutant, it's a very different story. First of all, you're seeing all these spikes, which we can quantify, and otherwise we're seeing periods where there's no activity. When we actually look at the frequency of activity in these low frequency to gamma profiles, it's basically blank. There's nothing going on. Now, if we go to higher frequencies between 200 and 600 hertz, different from here, which is 0 to 120 hertz, we do see some brief activities. And these are what we call high frequency oscillations or high frequency activity. And these particular kinds of activities or oscillations can actually be associated with pathological changes or epileptogenic tissue. So it makes sense in the mutant, in Rett patients who have a tendency to get epilepsy, you see high frequency activity, but not this lower frequency activity. We could do a lot of things with this model, but one of the things we did was to try and isolate why is this happening. Right? We're seeing these changes in physiology, and with the fusion, we can do this kind of mix and match experiment because we are, after all, generating the organoids, the cortex or the GE, which has the interneurons, the cortex, which has mostly excitatory cells, and then fusing them. So we can do this mix and match, quote unquote, experiments where we make hybrid fusion, where either the interneuron component primarily or the excitatory neuron component primarily is mutant and contains the Rett mutation. And so when we did this is the data you've seen. What do we see? So if we have the mutation primarily in the cortical component, in the cortex, excitatory cell component, it kind of looks like isogenic control. If we do the opposite mix and match, and the mutation is primarily in the ganglionic eminence component, then it looks a lot more like the mutant. So overall, what these data suggested was that in this phenotype that we're seeing, the striking phenotype of abnormality seems to be primarily coming from the GE component, suggesting an interneuronopathy, which is consistent with what's been shown previously from mouse studies in Rett syndrome. And so here, just to kind of circle and take your eyes to where we're seeing similarities between the two, right? So in the interest of time, I'll just run through this quickly, but essentially we came up with this model in Ben's lab with our collaborators, and we're able to see a specific deficit in this use case in the case of Rett organoids. And this is the structure. These are the approaches and techniques that I'm now using to move forward very briefly in terms of what I want to do in my own lab. So first, I'll just describe a specific project. So we don't just have to make cortex organoids. We can make other kinds of organoids. I've been making hippocampal organoids. And this is based on previous studies from Sakaguchi and in Ben's lab, actually from Momoko, who started this work. And we see unique kind of activity profiles in these hippocampal organoids. Now, this hippocampus isn't going to look like the ex vivo hippocampal slices that many of you may be used to. This is going to be different, but we at least want to make sure that we have the cell types that we'd expect to see in hippocampus, in our hippocampal organoids, and we do. So ZBTB20 and NRP2 are markers of hippocampal primordium, so developing hippocampal cells, which we see. PROX1 is for dentate granule cells, and we see some of those, and we see GLUC1 is a kinase receptor antibody, and that's usually associated with CA3-like regions, and we see those kinds of cells. And when we look at the oscillatory activity, importantly, we see oscillations. There seem to be some differences between cortex. In particular, we see more robust theta and alpha frequency oscillations. And we may also be, this is very preliminary, but we also be seeing some unique oscillations like this thing, which is a sharp wave ripple complex, that are relatively unique to hippocampus, and in this case associated, if they turn out to be real, with hippocampal memory consolidation or memory formation. So the hippocampus is behaving differently. These hippocampal organoids are behaving differently than cortex organoids. And so now moving forward, we want to characterize this, and we can use this to potentially study brain region differences in circuit activity, and also, of course, pathology in hippocampus from different, for example, genetic mutations or other mutations, or other pathologies, I should say. And finally, kind of more long-term in terms of my lab, I'm interested as a clinician in using the organoid system to also study patient-specific diseases. So I see patients who have these really severe developmental epileptic encephalopathies, this untreatable, rare, or poorly treatable, rare, very severe epilepsies. This is an example of a kind of stereotypical EEG that we see from some of these patients called a suppression burst pattern. And here is our calcium indicator imaging, not the same patient, different, like completely different patients, but the same named disease. And so the thought is, can we actually go directly from patient to organoid and model the disease for specific patients and organoids? And in this case, it makes sense because these are really rare diseases that are not easy to treat. Other examples of this, so that's from calcium indicator imaging, from LFP data, we get this kind of, these oscillations all the time. I've already shown that this is old data. I'm focusing here on delta and gamma oscillations. Well, my clinical colleagues in the pediatrics epilepsy department, Raj Rajaraman, Sean Hussain, and others, use this exact same, these exact same oscillations, for example, to look for responders versus non-responders to medications for really hard to treat pediatric epilepsies. So theoretically, we could do this, and this is sort of something that we want to pursue in more detail further down the line, basically making an organoid of getting iPSCs from a specific patient and trying to model that disease. So final slide, really in terms of my vision, how I think about organoids in my lab, I think of the organoid, warts and all the limitations. I understand those, but primarily as this tool to really unleash the potential of a stem cell in a way that other in vitro systems like 2D culture can't, so that we can study these sophisticated problems and learn something about neurological disease that maybe we couldn't using other models that exist. So the last thing, I really need to thank a lot of people, but in particular, I'll focus on Ben, who's my primary mentor, who's of course a brilliant scientist, but just the most generous and kind person, has helped me a lot, and Ishwan and Payman as well. One of the reasons I'm really excited to stay here is that these folks can hopefully still be my collaborators and we can continue this work. So thank you.
Thank you very much. Again, really beautiful work, and I think we all clearly see the future of where you're going. So this will be awesome. Again, so thank you, and I apologize, we have to move on. So please send an email directly to Ranmal if you have questions or want to collaborate with him. So that gets us to our next speaker of today, who is Ausaf Bari. Ausaf got his undergraduate degree from UC Berkeley, our second Berkeley person, and then went to Boston University to get his MD PhD. For his PhD, he worked with Chris Pierce, and then came back to UCLA, where he spent quite a long time in residencies and internships, so really did his clinical training here, working with many different people at UCLA, but especially Nader Pouratian. And he got his own lab now and is going to talk to us about pleasure, pain, and bionic brains. And he's actually the only one who's not from neurology. His appointment is in neurosurgery. So Ausaf, please.
Thank you, Dr. Schweitzer. Let me just start my presentation here. And thank you for giving me this opportunity to talk about my work. So I will be summarizing a lot of what my lab does right now, which is ultimately related to the neurosurgical restoration of nerve and brain function. And these are just my disclosures. So what, as a functional neurosurgeon, what I really do is specialize in invasive neuromodulation. And for most neurosurgeons in my line of work, what that really boils down to is something called deep brain stimulation, which is shown here on the left, which many of you are familiar with. But essentially, this is a source of electrical current and a way to use electrical neuromodulation to affect neural circuits in the brain. Currently, there are 5 FDA-approved DBS indications that are listed here. We are very familiar with some of these. Essential tremor, Parkinson's disease, and dystonia are the movement disorders that are on label. In addition, obsessive compulsive disorder is also FDA approved, and more recently, epilepsy or certain forms of epilepsy were approved. There are other forms of invasive neuromodulation, including this device here, which is the responsive neurostimulator used in epilepsy. So one of the major things that my lab is focused on and other people throughout the United States are trying to do is to apply deep brain stimulation for other disorders, particularly those that involve the affective circuits in the brain, such as depression, chronic pain, and addiction. And ultimately, as this technology matures, we'll be dealing with more of the ethics of using this to enhance function potentially in healthy individuals. So traditionally, DBS and other forms of invasive neuromodulation were used and are currently used for amelioration of motor disorders and motor circuits. And some of the reasons for that is that motor output is easily quantifiable, it's more objective, but more and more we're interested in using this technology to modulate these cognitive circuits and affective circuits. And for example, cognitive circuit modulation includes deep brain stimulation for things like dementia and Alzheimer's disease. And mood and affective neuromodulation includes addiction, chronic pain, and things like depression. So what my lab is currently working on is the study and mapping of human emotion. And that includes how we translate affect, pain, and reward into motor behavior. And Ahmet said that the brain exists to move. And I would argue that we move to produce positive emotion. And so really the brain exists for happiness ultimately. And so my lab is really studying how we're going to modulate these circuits to increase our levels of happiness and reward and decrease pain and suffering. And so there's a two-pronged approach to this. One is that we can use deep brain stimulation and simply characterize the behavioral changes in the patients that we have implanted. But the inverse approach, which I find even more interesting, is to take patients who are implanted and to elicit behavioral responses through task-based paradigms and to record simultaneous neuronal activity. And so we can do this intraoperatively using awake recordings, and that would include LFPs and single units, or we can do this extraoperatively, which is mainly LFP recording, and sometimes we can also do single unit recordings. But the overall goal is to understand the interplay between emotions and behavior, and specifically appetitive versus pain-related behavior. So this is an example of what we do in my lab. We focus on an approach of both invasive electrophysiology in awake human beings, and also combine this with probabilistic tractography and other advanced neuroimaging techniques to map these circuits. So you can see here, this is a patient in the lower part of the screen here. This is a patient undergoing DBS for Parkinson's disease. The patient is awake and performing motor tasks, which we do for clinical testing in the operating room. On the other side of this sterile barrier, we have the neurosurgeons operating and recording local field potential or microelectrode data directly from the basal ganglia. But we can modify these tasks as shown in the top video here, which is showing the same subject now performing a monetary reward task. So this patient is being asked to choose from different monetary values in the future or a present value, and we're simultaneously recording from both the cortical surface and the basal ganglia. And in addition to this, I'm using these advanced neuroimaging methods. This is an example of a brainstem atlas that my lab has just recently completed. This is an atlas of all of the relevant, or most of the relevant deep brain structures in the brainstem. So this is a brainstem atlas of human emotion that will be released very soon into the open source realm. And we can subsequently use this kind of technique to map out these white matter pathways that are relevant for different types of reward processing. So another example of using this is in our patients that are awake and are already implanted with deep brain electrodes for seizure monitoring. These patients spend 1 to 2 weeks on the ward, and they have up to 10 electrodes in various limbic areas of the brain. And one of the things that my lab is interested in is looking at this local field potential activity in the mesial temporal lobe and orbital frontal cortex in monetary reward. This is just an example of showing that we can predict based on the LFP activity what the patient will choose in terms of their choice. So we extended this technique in these patients. This is an example of a patient that's undergone the placement of 10 electrodes in his brain. This is actually a case I just did yesterday. And you can see all the various trajectories here. This is an example of a CT scan showing you these electrodes. This is an electrode in the orbital frontal cortex with the context spanning from the mesial to the lateral surface. And this is kind of what it looks like when it's hooked up to the recording equipment. So we're extending the LFP work here. This is an example subject showing single unit data from an awake human being doing a temporal discounting task. And we're again seeing that the firing rate here was correlating with the patient's choice. And this is very fresh data that we just collected over the last few weeks. And my lab is now analyzing this data from 10 different subjects. So we have over 100 units worth of data now from these patients. So that's all fine and good. We're using our existing techniques to map out these brain circuits, but we eventually want to develop novel therapeutics. And one of the areas that my lab is focused on is developing invasive neuromodulation for pain. This is an area that affects over 25 million adults with an enormous cost to society. It's actually chronic pain, especially chronic back pain, is the most common reason anyone will go to see a physician in their lifetime, and has contributed to the opioid epidemic. And so we're very interested in applying neuromodulation for chronic low back pain. And there's been this paradigm shift in dealing with this. So we've been doing this for several decades now in terms of doing DBS for pain, but we've been targeting the sensory circuits. But there's this shift in understanding of nociception as more of the sensory discriminatory pathway, whereas pain itself is more of a subjective and conscious perception, which really involves this interplay between cognitive domains, affective domains, and also the sensory domains. So in order to do this, we really built on other labs that have looked at fMRI data that showed that when we look at chronic back pain patients and look at the spontaneous fluctuations in their pain, it really activates these frontal, emotional, cognitive, or affective areas. Whereas either the same patient experiencing acute nociceptive pain or normal controls will activate the traditional nociceptive pathways. So this is not not really new information for neurosurgeons. So this is a slide from the 1960s for a procedure called an anterior cingulotomy, in which neurosurgeons would go in and actually disrupt the cingulum bundle, which is a fiber pathway that's interconnecting these affective areas. And when you do this, interestingly, what happens is the patient will tell you that they still feel the pain. They can still feel the sensation of pain, but what's different after the surgery is that it no longer bothers the patient. So you've disrupted or disconnected affect from sensation. And so we still use this technique in neurosurgery. I still do this here. We reserve this for people that have a very high suffering component of the pain. It tends to be patients with cancer pain that have a short life expectancy. And the technique is a lot more advanced now. We use stereotactic navigation. We use a very minimal opening with a radio frequency ablation probe, and we can use MR thermometry to guide our ablation. And so in terms of applying DBS for chronic pain states, this is a state space model of pain in which we hypothesize that neuronal activity in these circuits, in these affective, cognitive, and sensory circuits, can contribute to a pain state. And the goal of DBS is to push this activity into a pain-free activity state and to maintain this long-term using things like closed loop or sensing DBS. And so we hypothesize to target an area of the brain called the subgenual cingulate cortex. And the reason we chose this area is because this has shown a lot of efficacy in deep brain stimulation for depression. This is an area that is hyperactive when people have sadness, Even normal healthy volunteers, when you do PET studies on patients that are recalling sad autobiographical memories, will show activity in this area or increased uptake in this area. And so this area has been already targeted by neurosurgeons for treatment for depression. And neuroimaging studies have shown that this area is highly connected to the orbitofrontal cortex, the cingulate gyrus, and these mesial temporal structures that are involved in affect. So some of the work done by a former postdoc in my lab used probabilistic tractography to map these circuits and showed that this area was highly connected to the ventral striatum, the anterior cingulate area, as well as the medial prefrontal cortex. And this kind of formed the basis of an NIH grant that we applied to, in which we proposed to use directional DBS, which is a more modern form of DBS that has the ability to selectively engage various white matter pathways using this kind of directional stimulation capability. And we applied and received this $4 million grant from the NIH under the HEAL initiative, which is the Helping to End Addiction Long-Term initiative by the NIH. And this will be a first-in-human feasibility study in which we'll be implanting this device in patients with chronic refractory low back pain. And the patients will be prospectively randomized to either sham or active stimulation. And our aims are to show first feasibility and preliminary safety. That's the major goal of this application. And secondarily, to show preliminary efficacy. And our third aim is to use probabilistic tractography to optimize the response in those patients that we expect will not respond to the therapy. Because we have a hypothesis that there's going to be a fingerprint that we will identify that will correspond to actual responders, and we'll be able to push the non-responders to the responder fingerprint using this combination of probabilistic tractography combined with deep brain stimulation. So that was kind of a whirlwind overview. I know we were short on time, so I wanted to give you a broad overview of what we do. These are some of the active, some of the areas that we're doing is using these modern devices. So there's a Medtronic Percept device. This is an interesting device, which actually allows you to implant a pulse generator in a patient that undergoes deep brain stimulation for something like Parkinson's disease or tremor or pain. But we can bring the patient back to clinic and perform chronic ambulatory LFP recordings from the human brain. So we are now using this to study these chronic pain circuits, but the patients are also performing reward tasks and addiction tasks to understand the role of the basal ganglia, especially the ventral basal ganglia, in these pathways. So we use this device now. We've already implanted probably 20 patients with this device. And this is an existing device, which is the RNS device, which can also perform ambulatory long-term recordings from the mesial temporal lobe in patients with epilepsy. We're also collaborating with Dr. Anderson's lab at Caltech to develop BCIs for motor control in tetraplegic patients. And finally, I'm actively working with Dr. Babakani's lab in the UCLA bioengineering group to develop modern wireless DBS devices to provide future wireless DBS. So with that, these are my acknowledgements. And again, you can reach me offline, and we're very happy to collaborate with all of you. Thank you.
Thank you very much for that whirlwind going through the basic explanations and the applications. Very, very wonderful. And I guess we'll just have to have you back in 3 months and see, where your study is happening, where you're going with this. So that's really awesome. Thank you. And last but not least, we have Steve Cramer, who got his undergraduate from Berkeley, and then did his MD at the University of Southern California, and came to UCLA for first residency, and then did a lot of his additional training at Mass General in Boston. He's now full professor at UCLA and is the director of research at the California Rehabilitation Institute. And I'm really sorry that maybe some of you have to leave. We're running a little bit behind time. I apologize to all of the speakers that I had to rush you. So Stephen Cramer, thank you.
Thank you, Felix. Can you see my single slide okay? Sometimes things.
We now see double slides.
You see double slides?
Yeah, we see your presenter view. The other side, you might have to share the other screen.
All right. Try that one.
Yes.
Better?
Yep.
Great. Well, thanks for having me. My name's Steve Cramer. I'm a stroke neurologist, and I focus on stroke recovery with an emphasis on the motor system. Like so. In my lab, we say, no movement, no Mozart, no Monet. These are my disclosures. And I'll try and make this quick, given the remaining time. These are the main points. Briefly, we'll do some introduction, reminding you that stroke is amongst the leading cause of disability in our species perennially, and that motor deficits are probably the most common form of deficit leading to stroke-related disability. There are emerging restorative therapies, therapies that promote plasticity and rebuilding from elements that survived the initial stroke, emerging therapies in preclinical data, and translational efforts to see what we can do in humans. The problem is getting the right patients collected to the right model, and we'll get into that, which will be the main part of this brief talk. Stroke happens, there's death of tissue, this is followed by inflammation, and a whole cascade of growth-related events occur bilaterally after a unilateral injury. Animal research is taking advantage of this knowledge and examining therapies that respond to this biology. So we don't have time to get deep into it, but vagus nerve stimulation, growth factor injection, work from Tom Carmichael's lab, Ahmet, CCR5 inhibitor. There's a certain form of bone marrow adult cell that may be useful, enriched rehab. All of these things have in common that they can be initiated not hours, but days after the acute injury, and are associated with sustained significant behavioral gains as far out as people have looked. This is one review I wrote trying to say, well, where are these things being translated? Where are they in the progression? And activity-based therapies that are less based on animal work are most ready for broad clinical application. And many of the biologicals are still the most in preclinical development and different categories of therapy at different stages. I'll tell you about two studies, but then I want to get to the good part. So vagus nerve stimulation is of some interest. The vagus nerve collects afferent data from throughout the body, reports it to the cortex through the brainstem, and results in release of norepi and acetylcholine, and is a strong modulator of cortical plasticity. So I've been working with Michael Kilgard and the company that he spawned for 11 years now. They had some early animal studies showing increased cortical plasticity with vagal nerve stimulation. And they did 3 studies in animals showing that if you stimulate the vagus nerve during movement after a stroke, you get better motor outcomes. Briefly, 7 days after stroke, rats got vagus nerve stimulation versus sham. You pair the stimulation with successful trials. Here's the stimulator, and here are the successful movements. And what you see is significant behavioral benefit in animals that is significant, generalizes to a different task, and is sustained. So they did 3 preclinical studies, and I helped them write their first studies. And we recently published our 3rd study, which was a double-blind, sham-controlled randomized trial of 108 patients with chronic stroke. They all got a VNS implanted, a little cuff in the neck. On half the people, we turned it on during rehab, and half the people, it was not turned on during rehab. The idea is that you have to pair activity with stimulation to optimize the plasticity benefit, experience-dependent plasticity. They did this in the clinic and then at home. This is what the system looks like. The patient makes a movement and the therapist stimulates the vagus nerve with each movement. And bottom line was we saw significant results. This is a modest but statistically significant benefit on the primary endpoint in the red box, but I'm more intrigued by secondary endpoint in the green box. This is the fraction of people who had a clinically important response. You can see that with vagus nerve stimulation, it's about half, and that with no stimulation, just the therapy, it's about a quarter. So that's great. There's a doubling of the rate of responders. On the other hand, only half the people responded. Why is that? That's what we're going to get to in a minute after we look at one other therapy. This is a telerehab system. I don't have time to get into it, but people have motor deficits, very common after stroke, as I said, and they want to have more therapy. They want lots and lots of activity. Animal studies show, work from Randy Nudo and others, you probably need 700 to 900 motor repetitions a day to reach maximal motor system plasticity. But in human practice, it tends to be more like 20 to 30 repetitions per day because modern clinical rehabilitation is not built to deliver high intensity motor rehab. So we reasoned we could put something in a home with lots of transducers focusing on arm motor, and we are able to get people to do over 1,000 arm repetitions per day. It may look like whack-a-mole, which is fun, but we are measuring movement at each of the different joints, the number of repetitions per day, and documenting over 1,000 repetitions per day for 6 consecutive weeks. And don't have time to get into it, but we did a national trial of this therapy as a standalone and saw very significant benefits that were equivalent to what you get people going to clinic every day. Okay, but the key questions are, why do only some people respond? What is it about the brain that makes it more likely to respond to a given therapy or less likely? What is the biological model for the treatment intervention? And what can we measure in humans that tell us whether that person's biology is aligned with the model or not? And we can use behavioral status, but don't have time to get into it much today. But final behavioral status, the behavioral phenotype that we measure at the bedside is at best a rough marker of the underlying neural state, neurophysiology, neural injury state. And so while behavior often does predict treatment response, it tends to be a rather poor predictor. And what we're seeing, and this is bottom line for the remaining couple of minutes, is that measures of neural injury and neural function tend to be much better predictors. Why? Because the word stroke comes from the Old English word for strake. If I said to you, how do you treat somebody who's had a strake? You'd say that's silly. You say, how do you treat somebody who's had a stroke? It's not much less silly. Been talking about stratifying for some time now. We measure corticospinal tract injury. We use diffusion tensor imaging tractography in healthy subjects to map out the normal corticospinal tract. And then we superimpose each lesion on it and come up with the percent corticospinal tract injury. X-axis is percent corticospinal tract injury affected by the stroke. And you see that the higher the injury to CST, the smaller the gains from 24 patients getting daily intensive robotic therapy. So that's a significant relationship. CST explains who's going to respond, explains about R squared about a third of the variance. We can explain about a third of the variance with anatomical measures, but there must be more to it in terms of why some people respond and some don't. And in another study, 45 patients, odds ratio of 15 when they have severe injury by this measure, just validating the measure of CST injury. So it turns out, as you may know, neural function and neural injury are not 100% correlated. There are differences in the function of cortical networks, cortical subcortical networks that are not entirely captured by anatomical measures. And so we rely on measures of neural function to further understand the state of the brain and further try to understand whether that brain state is aligned with the model for treatment efficacy for people who have motor deficits after stroke. Our initial studies of neural function relied on resting state fMRI. And so in this study of 30 people who got intensive arm therapy with a robot, we see that the more that the ipsilesional motor cortex shows functional connectivity with contralesional motor cortex, the higher the gains were from therapy. And so putting a seat at the table for functional measures. And in the same study, the percent CST injury once again correlated with lower degree of gains. These two measures were not correlated, and together they explained about half of the variance in treatment response. So functional measures have a seat at the table, but we're still not there all the way. Part of the problem is that MRI is a complicated thing to do. As Felix kindly mentioned, I'm the director of research at California Rehab Institute. It's one of the largest rehab hospitals in the country. It's about 2 miles away in Century City. We have a lot of patients who stay here for weeks at a time. Come on by, give me a holler. We'll try and find a research ground in common. So I can't just take people and wheel them over to the MRI scanner at UCLA. So we're looking for ways to measure brain function in a more accessible way. Fortunately, I've been doing dense array EEG studies for the last few years in anticipation of this. This is the EGI system we use, 256 leads. It's saline based, impedances are a little higher, but the waveforms are perfectly fine for our purposes. In one of our tele-rehab studies, patients got intense therapy for 4 weeks, and we asked which forms of high beta range connectivity predict future motor gains. And it turns out that the PLS map, partially squares map, a validated model, shows us that high beta connectivity with ipsilesional motor cortex predicts about 61% of the variance in treatment response. And one of the main areas of connectivity was ipsilesional to contralesional motor cortex. It's very clear that the two hemispheres interact in a way that's very important to outcomes after stroke and important to responsiveness to therapy. We've looked further at this. We looked at the anatomy of the motor section of corpus callosum and showed that it has a lot to do with behavioral outcomes after stroke, but particularly in subgroups. So one of the key messages as we finish up here is when measuring biomarkers that correspond to biological state, biomarkers that predict treatment response for restorative therapies in patients who've had a stroke, it's not one size fits all, and different biomarkers are likely to be more or less useful depending on different features of the patient. In this case, we found that the larger the stroke, the more severe the deficit, the more motor subsection of corpus callosum, functional fractional isotropy, white matter integrity, was a better predictor of clinical state. In people who were more severe, corpus callosum was less relevant. Okay. We also look at genetics. I don't have time to get into it, but we have found a polygene risk score in the dopamine system models the system well and predicts behavioral gains. And this has been validated independently in 3 labs around the country. Last slide, we followed 750 patients with stroke for a year at 30 sites across the US, looking at a number of classes of outcomes, mood, anxiety, function, motor, social status, et cetera. And for each, we have specific polymorphisms that we hypothesize will be related to outcomes in those domains. We're also looking at some polygenic risk scores, and we'll look at GWAS, but we're probably not powered for that. So the main points are that stroke, common cause of disability, especially with motor deficits. The therapies emerging from animals can be translated to humans, but we really have to understand the biology of the human brain in order to match the right patients with the right therapies. And with that, I thank you for your time today.